In this Jupyter Notebook slides that you see here, we will talk about Sub-SBML Python toolbox. It's a Python toolbox for modeling diffusion across membranes and compartmentalized systems. As I already mentioned, if you do not have the software already, you could just do pip install Sub-SBML, and that should get the software installed on your computer. Also, an important point here is that you have some sort of SBML simulator in your computer already installed. The preferred choice that I will be going with today is Bioscrape. It's a fast simulator and it allows SBML models to be simulated. Also, to create your models, to create the SBML files, you would need your choice of a model builder tool, and we will be using BioCRNpyler to do that. Some quick notes before we jump into the code. My biggest debugging advice while working with Sub-SBML is read your SBML model output to find whatever bugs that you think are occurring. So to do that, just click on the XML file, the SBML file with extension .xml, double click it and just look through it. And slowly you will get hang of what an SBML file looks like, and that's very helpful when debugging your models. The second note is on name and identifier of a species or of a reaction or a compartment in an SBML file. And this is related to the question earlier. So SBML has, all SBML elements can have a name and an identifier. And as you can imagine, name is something which is more of a descriptive name that you give to your element, to your species, reactions, compartments. An identifier is something that you specify that the software interfaces with. So usually the simulator software or the model building software, they have to make sure that they are creating the correct identifier for a species. But Sub-SBML, to find out what species need to be combined together, works with both the name and the identifier. So if two species have the same name, they get combined together. Identifiers are always unique. Anything that gets combined together in Sub-SBML has to have a same name. So let's go through a simple Sub-SBML use case where we combine two different models. So what we will do is we will have a model of transcription and another model of translation. If put together, we would have, starting with some initial concentration of the gene, we will have protein output. But if put in separate compartments, just as a hypothetical scenario, we would not have the protein output be produced unless we combine them together correctly. And we will demonstrate that using Sub-SBML. So for the transcription model, I'm simply taking one-step transcription where the gene G transcribes with some rate that is dependent on the inducer signal S to produce the transcript T. And the second reaction is just the degradation of the transcript T. And the translation model is also a one-step model where the transcript T gets converted to the protein X. And then the protein X degrades at a rate of delta. So once we create these models and store them in an SBML file, we can use Sub-SBML to combine those together. So how to do that? First, we start with simple import statements. We import the subsystem creating functions like this here. So let's go through this line by line now. So the first line is create subsystem, as the name suggests. This function is the function that you will use to import SBML files into your model and create a subsystem for it. So the file that I have already created that I will go through a bit later is the transcription SBML model. And this is an XML file. So this is the SBML file that I've created. And I'm importing it to create subsystem SS1. And I can give a subsystem name. So what this does is it appends all of the elements in this file with this name so that it's a specific subsystem now with this name. And similarly, I create another subsystem for translation. And this is done by importing another SBML file. So now let's very quickly look at how we might generate, like where do these come from? Because that's not covered here. It's like these files have to exist. So I have shared 2 notebooks, and in the second notebook that's called Generate Models for Sub-SBML, you will see all of the model generation steps here. So the transcription model, as I mentioned, can be generated using BioCRNpyler, and it's a simple model with species I'm calling G, and I'm calling it now mRNA_T. That's my transcript name. And this is the creation of all the reactions. And then we write an SBML file with this name. Similarly, we create a translation model with transcript T, protein X, and then we output the SBML file translation_SBML_model. And these are the 2 files that I'm importing here to create the 2 subsystems. Now, if you've observed, in the first subsystem, I'm calling the transcript mRNA_T, and in the second one, I'm calling it T. If I want this combination to work properly, I would need to rename the mRNA-T in the first subsystem to T. And this is a utility function in Sub-SBML that you can use to do that. So we call rename sName and rename the mRNA_T with T, and then we can combine together to see if it works. Another helpful function here is set species amount. So I'm setting this initial condition for the gene G to 1. And then combining the 2 subsystems is as easy as just writing SS1 plus SS2. So this plus operator works on the subsystem objects that we've created and then gives out TXTL subsystem. It's a new subsystem that has the combined model for transcription and translation together. There's a longer way to do the same thing is if you create a new subsystem, which is an empty placeholder, and then you call the combined subsystems function, give a list of subsystems that need to be combined. Usually, this is the way to go when you have more than 2 subsystems. And this gives you the combined subsystem in this subsystem object. And finally, once we have this combined subsystem object, we can write it to an SBML file, as in this line. And that's it. We can run the simulation now to see if it worked. So this is just some simulation code, and I am plotting the protein dynamics for different values of S, which was the signal inducer. And as we can see, it's working with the protein concentration going up. If you just had the translation model on its own, this would not be the response, and that's an exercise that you can try. Next is, if you remember, the overall Sub-SBML framework is that you can create models for systems with membranes and signals being transported in and out of the membranes. And you can combine all of that together to create a combined system model. So let's see how that works out in with an example. Now I have third subsystem for the signal induction. And so I'm just taking a very simple model here where a signal gets produced in 1 step, some rate. And this model, again, how to generate it, we go back to the generate models for Sub-SBML notebook. And here we have the signal induction model. We use BioCRNpyler again to create the species and the reactions, and then most importantly, write the model in SBML file. And I'm calling it signalinmixture.xml. And that is exactly the name that I will import here to create the subsystem. So this is the file that I've created using my other notebook that generates the SBML model. Then I import it into Sub-SBML by calling create subsystem. And now I want to obtain the combined subsystem with the signal as well being produced, and then the transcription, and then the translation. So like we had previously. So this is the third subsystem. So we combine all of these together by using combine subsystems function. This is the single most important function in Sub-SBML because it lets you combine subsystems together, and it takes care of everything that has the same name being combined together and so on. So we store this in this combined SS subsystem object, and once we run this, we have the subsystem object with the combined model together in this object, and now we can write it to an SBML file and give it any name. So that will be our SBML file output. We can run simulations again to check if everything worked, and this is just some simulation code. And now we are seeing with different inducer concentration, the signal gets produced, and then the signal activates the transcription, and then the mRNA translates to form the protein X, and that's what I am plotting here. When all of this was happening, it's important to know what exactly is going on underneath. So for compartments, if two compartments of the same name, they are combined together if they also have the same size. For species, and this is probably the most important, if they have the same name, they are combined together as well. And the important point here is that you need to make sure that the initial amount is set later to correct value. So if you saw here, I set the species amount after combining everything. I set the initial conditions so that just to make sure I have correct initial conditions in the combined model. Similarly, for parameters, they are combined if they have the same name and also the same value. So Sub-SBML is looking through all of these SBML elements and combining them together if they are actually the same. So it's making sure that there are no duplicates to avoid any kinds of bugs while running the simulation. And we saw a couple of utility functions that we used for subsystem objects, like rename sname to rename some species name. There are other utility functions as well that could be used. So for example, if you pass a verbose keyword argument while combining the subsystem, it lists out all the warnings and prints out various messages about what it's doing with your models. So if you're very careful with what's happening with your models, you should probably be using the verbose keyword to be set to true when combining the subsystems. There is also a create basic subsystem function that can give you a basic SBML model to start with. There is another function that can, and this is another important function, to get species by name. So as I mentioned earlier, species have a name and an identifier. So a name is something that we as humans understand, and identifier is something for the software. So get species by name can look at the species list and give you out species identifiers or objects by the name that you have specified. There are more utility functions to help with, and we will probably be using those in the examples next. So this is the final example, which is the most interesting one as well, where we have a system with some subsystem models inside, and it has a membrane where some external species gets transported into the membrane. So let's see how we model this using Sub-SBML. in particular, the system that I am showing here is a system with transcription and translation as subsystems in the internal of the system. And there is an IPTG reservoir in the external. So there is a lot of it in the external subsystem, and it has to diffuse in to this system. But for the very simple example, for system 1, there is no membrane. So the TXTL subsystem inside can directly access this IPTG reservoir on the outside. So to create this system model, we use the Sub-SBML constructor of a system. So I create a cell called cell1, cell_1, and that's a system object. So a system object can contain various subsystems inside it. And we will see how that works. So we create now 2 new subsystems. So the first one is the TXTL subsystem that I want to put inside my cell. The second one is the IPTG reservoir that is supposedly outside of the cell, but right now in this example, there is no membrane. So this will also, these are all just together. And I'm just renaming something, so just to make sure they are having the same name. And if you notice, this TXTL_SS is actually the combined model that we obtained earlier. We combined the transcription and translation model, and now we are using that combined model to create a new subsystem inside this cell that we have created. And so now the 3 most important functions while creating models like this are here. So the first one is set internal for the cell object. So I have this cell1, which is a system, and set internal specifies what are the subsystems that are inside this system. So in this case, SS1 is the subsystem that is inside. Set external is this command to specify what's outside the system. And so in this case, SS2, which is the IPTG reservoir, is outside the system. The third one, which is commented out right now, is the set membrane function. And this function sets the membrane for this cell, and this is commented out right now because there is no membrane for this example. So set membrane sets the model for what the membrane is like, which would consist of all the transport reactions, and we will look at in more detail in the next example. And then finally, we can set some initial concentration, and to get the combined model for this whole system that we have created, we do cell1.getModel. And what this does is it goes in and looks at all the subsystems that are inside, combines them together, all the subsystems that are outside, combines them together, and then finally combines all of these with correct compartments into one SBML file, so that you can simulate this one SBML file that has different compartments, internal, external, and with transport reactions that might be occurring between these compartments, and give you out this combined model. And I can write it to an SBML file like this again, similarly, and that's it. Now we can simulate this SBML file to see how it's working. So we will get to the simulation part, but before that, create another system, but now with a simple membrane. So now we will do exactly the same thing. There is a TXTL subsystem inside of a cell, and there is IPTG on the outside, and it needs to transport in through a membrane. So for this, we will need to create a membrane model. So the first 3 lines are exactly same as before. We create subsystems for the TXTL, for the reservoir, which is on the outside. But now we also need a membrane model. So for this, things get a little bit more special, where in create subsystem, I specify that this is a membrane model so that it handles it accordingly. And why is this special? Is because a membrane model is always something that has transport reactions in it. So it has reactions between two compartments. And this is the membrane model that I've created that is being imported in here as the membrane for this system. So let's look at how the membrane model is created. So we go back to the generate SBML model notebook again. And here you will find IPTG simple membrane model. So for my simple membrane, I have decided that I just want this one reversible reaction that models IPTG going in and out. So there is one species IPTG in the internal compartment and the same species in the external compartment, and one reversible reaction for my membrane diffusion model. So to create this, since we have multiple compartments now, we cannot use BioCRNpyler anymore. And so instead, Sub-SBML provides natively some basic model creation tools as well. And we will demonstrate some of those tools while creating this membrane model here. So after importing, so simple model and simple reaction, these are the two classes that we will use to create this model with multiple compartments. So start with the first line here is just creating a new subsystem. This is just an empty subsystem. Then we create a new model inside the subsystem with appropriate units, and then create more unit definitions as required. Then the two most important lines here of the code are here that create the two different compartments of the membrane model. And now this is important to understand because, as I mentioned earlier, a membrane model in Sub-SBML is something that models the transport of species. And so to model transport of species across compartments, you necessarily have to have two compartments. So a membrane is always the boundary between two compartments in this framework. So a membrane model has to have two different compartments with two different names. And so I'm creating a new compartment. This is a Sub-SBML function for this IPTG MB. This is the simple model object that I've created to create this model. And I'm giving the name for this compartment as internal, and for the second one, I'm giving the name external. And I can give the size for these compartments as well. Could be anything. And then the units. So I have now in this model two compartments, and now I can create species in different compartments. So for example, the first line here says, create new species called IPTG in the external environment. The second says, create new species called IPTG in the internal environment. And so now we have the same species with the same name, but in two different compartments. So that's fine. And once we have that, we want to create this reaction, this reversible reaction that models the transport. So to do that, we first create the reaction rate parameters, so the binding parameter and the dissociation parameter, KB and KD. We then create a reaction between IPTG external and IPTG internal. So now, since they both have the same name, as I mentioned earlier, the identifiers always have to be unique. And so to do that, what I do is I look into the SBML file and see what is the unique identifier for one of these, because one of these has to have something different. And I find out that it's called IPTG_1. And so in this reaction, when I'm creating the reaction, I just give some ID to the reaction, R1, set reversible to true, and then create new reactant. So the first one is IPTG, which is the external IPTG. So I created this first, right? And then the second species that I created was called IPTG again. So that was edited internally for the identifier. So that's something that the users do not have to know. But for this case, since we want to create reactions between these two species, we create for this particular identifier IPTG_1. That's our product. and it's a reversible reaction, and then we create the rate for this. And finally, write it to an SBML file that we then import when creating the system. Going back, we create a subsystem for this membrane that we just created. And now we use our three functions again. We create a cell2 object. That's a new system, cell2. And now internal is at SS1, external is SS2, like before. But now there is also set membrane, and the membrane is MB2. And I write the combined model for these into an SBML file. So get the model and then write it out. Let's go do another example, which is an even more complicated version of this. So now system 3 is the same thing, but with a more detailed membrane diffusion model. So the membrane model that I'm proposing here is that IPTG binds to a transport protein, forms a complex, and then this complex causes the diffusion. So I will not go into the details for the membrane model. It's exactly the same, how to create the membrane model, but instead of one reaction, we have two reactions here, where IPTG external binds to the transport protein, creates this complex, and then it forms IPTG internal. So same process, we use the same thing to create two different reactions this time and create the membrane model. Once we have that, we import the membrane model as a new subsystem called MB3, and then I create cell 3, which is the third version of this cell with a different membrane. So the first one was with no membrane, the second one was a simple membrane, and the third one has this more complicated facilitated diffusion model. So in the system constructor, so this is another way to do the same thing that we were doing earlier. So instead of using functions like set internal, set external, we are using the system constructor to specify the list of internal subsystems, the list of external subsystems, and the list of membrane subsystems. And also note that this points to the fact that you can have multiple membrane models together as well. So you could choose to have one membrane model each for a particular kind of transport. So maybe one membrane model allows only for the transport of IPTG, and then you want to have another membrane model that allows for the transport of some other species. So you can have multiple of those as well, and specify all of those together here in this list. And same thing as before, we create the model, combine, everything gets combined together by using this function, and then we write the SBML file to a new file, cell3._model. So finally, let's see how the simulations, what the simulation shows, and we can look at this simulation plot in the actual notebook. So what we see here is the first plot is just showing a zoomed in version for a shorter time period. So with no membrane, the simple membrane is the orange one, and the green is the advanced membrane. We see that the advanced membrane that we had was a two-step membrane. The response is much slower to start with compared to the no membrane and the simple membrane. So the no membrane was, there is no membrane, so of course everything happens instantaneously. The simple membrane, there is a reversible reaction, so it takes time to build up the IPTG. And then the advanced membrane, it needs to bind to a complex and then get transported in. And then in the second plot is just a longer time period. It compares how the three membrane models that we created with three different kinds of diffusion compare together. So we see that the simple membrane and the no membrane case are more or less on top of each other, but they are exactly not the same because the initial response was different. And then the advanced membrane is much slower, but catches up at steady state at the end. So with that, I would like to conclude with some pointers to some additional tools that are available with Sub-SBML that you can work with. And one in particular that I can point out here is combined systems. So this is something where you can use to combine the systems that we created together. So a system is an object that has a membrane, that has subsystems inside and outside. And now you can take systems like this and combine those together to get an even bigger model. And so you can do that as well using Sub-SBML. And finally, there are some things to try. You can use the notebooks that I shared already, and you can work on your own SBML model and try to compartmentalize it, and then use the combine subsystems function to combine those together. A second thing to try, slightly more interesting, is to implement an advanced diffusion model. So now instead of the one-step facilitated diffusion, implement a two-step or three-step model and run exactly the same simulation and see what changes. Final advanced thing, or a much more interesting thing to try, would be to do the following. That there is a subsystem that creates a protein called X. X then transports out of this through a membrane and then diffuses into a new system, which is could be an E. coli cell model. And then in the E. coli, this X activates a new circuit that produces a desired protein output Y, which is some other protein circuit. So X activates that and produces Y. And so you can model this whole process where X gets produced, gets transported out, then gets transported in, activates another circuit, and produces another protein. We can do this using Sub-SBML.